Good afternoon and welcome to all. Today we are going to discuss about historical perspectives. So today we can discuss what are the different historical events that have been taken place. People had no real understanding of why disease had occurred in earlier times. At that time, as a result, the civilization progressed. The people started clustering into cities. The crowding increased, and the spread of waterborne, insect-borne, as well as the skin to skin infectious disease also increased a lot. Then, about the during the war, the infectious disease was always intimately concerned because of the poor sanitation, movement of the people, and the malnutrition that a war begins. At that time, also, it causes a great increase in the bacterial infections. And in the earlier time, the first division aid was invented. It was the inventor is unknown. It could be possibly a monk. So he invented a reading stone around 1000 AD. It was a glass sphere that magnified when laid on the top of the reading materials. In the year 1590, the two Dutch eyeglass makers, Zacharias Janssen and the son Hans Janssen, experimented with multiple lenses placed in a tube. The Jansons observed that viewed objects in front of the tube appeared greatly enlarged, creating both the forerunner of the compound as well as the telescope. It led to the foundation of the discovery of both compound microscope as well as telescope. Then coming to the earlier discoveries, Lucretius, a Roman philosopher, and the Girolamo Fracastoro, a physician who believed that invisible creatures were responsible for diseases. Then another person, Francesco Stelluti, observed bees and weevils using a microscope in the early 1600s. Then the other most important person, called Robert Hooke, a young English scientist, became the first person to view and describe fungi using a simple compound microscope. In 1665, Hooke published his findings in a book called Micrographia, which detailed his observations of tiny cork like cells resembling little boxes. Hooke is known principally for his law of elasticity, which is also known as Hooke's law, and for his work as the father of microscopy. It was Hooke who coined the term cell to describe the small basic unit of cells. Next is the Hooke's microscope, which he used in the early times to observe those cells. Next is the another important person, Antony van Leeuwenhoek. He was a Dutch merchant, and his hobby was to make small handheld microscopes. Squinting through the lens at the specimen held on a pin, he discovered a world of invisible creatures, and he called them as animalcules, which is also known as small animals. He found them almost everywhere he looked. For example, in the water droplets, in the soil particles, and also even in the teeth scrapings. In 1674, Leeuwenhoek communicated his discoveries to the Royal Society of London, sending detailed drawings. His primary observations shook the scientific community, and this event led to the expanded uses of microscopy as a standard scientific tool. All his drawings and 9 of the estimated 500 microscopes that the Leeuwenhoek made still exist now. Also, the most powerful of those has a magnification of 266, the powerful enough to magnify an average sized bacterial cell to the size of the period at the end of this sentence. And this was the microscope which Leeuwenhoek used in the earlier times. Next is about spontaneous generation. This idea was based on that life routinely arises from a non life, which means it was a belief that life could originate from the non living or decomposing matter. And this idea was supported by the persons such as Aristotle, John Needham, etc. Aristotle, according to him, it was readily observable that aphids arise from the dew which falls on the plants, fleas from the putrid matter, mice from the dainty hay. This was all his beliefs, and these beliefs remained unchallenged for more than 2000 years. From the ancient times, spontaneous generation was thought to be the origin of many organisms, such as rats and flies, that routinely appeared in certain materials. Microorganisms always appeared suddenly in certain materials that had previously been free of them. These are all based on the observations in spontaneous generation. It seemed logical that microbes were the products of spontaneous generation. And for the 80 years, this above debate continued. Then the proponents of spontaneous generation seemed to gain ground, and when in 1745 an English clergyman named John Needham did a well publicized experiment. Everyone knew boiling killed microorganisms. Therefore, he boiled chicken broth, put it in a flask, and sealed it. Microorganisms develop in it only by spontaneous generation. So, he was the greatest supporter of spontaneous generation. Experiments with gravy seem to show that life could be generated from non living materials. This was the observations of John Needham. But there was an Italian scientist, priest, not scientist, and a professor named Lazzaro Spallanzani was not confined with the idea of spontaneous generation. According to him, perhaps microorganisms entered the broth after boiling, but it could be before sealing. Therefore, Spallanzani also performed certain experiments. He put the broth in a flask, sealed it, and then boiled it. No microorganisms appeared in the cooked broth. Still, the critics were not persuaded. Spallanzani didn't disprove the spontaneous generation. They said he just proved that spontaneous generation required air. Then another scientist called Felix Pauchet proved growth without contamination from the air. Then gradually what happened? This spontaneous generation was rejected as the origin of visible organisms. Another scientist, Francisco Redi, a physician in Italy, played a major role. In 1665, he did experiments with covered and uncovered jar of meat. He showed that maggots, which are called fly larvae, developed only in the meat, that flies could reach to lay eggs on them. Apparently, spontaneous generation did not occur, at least in the case of flies. Instead, the flies and by extension all the living things come only from the pre existing living things. So, it was the most important finding that the living things comes only from the pre existing living things. So, this was a greater controversy for the spontaneous generation. Still, many people believed that microorganisms were an exception to this rule. So, the spontaneous generation gained momentum during the late 18th and 19th century, and gradually this theory was disproved. John Tyndall, he also explained to disprove the spontaneous generation that omission of dust lead to no growth. He also demonstrated the heat resistant forms of bacteria. Next, about fermentation. It was the another important contribution of the scientist called Louis Pasteur. Before that, I am describing about the Louis Pasteur. He was a young French chemist entered to counter the argument that air was necessary for spontaneous generation. And the Pasteur disproved what the theory of spontaneous generation through his famous experiment that is called Swan Neck experiment. In the Swan Neck experiment, Pasteur boiled meat broth in a flask and then drew out and curved the neck of the flask in a flame in the shape of a swan neck. That's why the name Swan Neck experiment. No microorganisms grew in the flask. But when he tilted the flask so, some broth flowed into the curved neck and then tilted it back so the broth was returned to the base of the flask. The broth quickly became cloudy with the growth of microbial cells. Gravity had caused the microbial cells that had entered the flask to settle at the low point of the neck. They never reached the broth in the base until they were washed into it. Thus, the pastor convinced the scientific world that spontaneous generation of microorganisms does not occur even in the presence of air. So, Pasteur, through his simple but elegant experiments, grounded microbiology in scientific reality. Thus, he disproved the spontaneous generation. And also, another important finding of Pasteur was fermentation. Sorry for the disturbance. I am taking the uh, Pasteur slide. Another important contribution of the Pasteur was the defining fermentation. The Pasteur was able to prove that living cells, that is, the yeast, were responsible for forming alcohol from sugar, and that contaminating the microorganisms found in the ordinary air could turn the ferment sour. So, the yeast is responsible for forming the alcohol from the sugar, but the other microorganisms are important in getting causing the spoilage of this alcohol also. Next is about the diseases which are associated. I think the slide is getting problem. Mhm. Sorry for the inconvenience. Okay. Demonstration that microorganisms can cause disease. So, in this slide, we can demonstrate the microorganisms which are responsible for causing diseases through the eminent scientist. Agostino Bassi showed that a silkworm disease was caused by a fungus. And then another scientist called M.G. Berkeley demonstrated that the great blight of potato, it is an infection which occurs in the potato, of Ireland, which was caused by a fungus. The next is about Louis Pasteur. He showed that the peregrine disease of silkworms. This was caused by a protozoan parasite. Then he also developed the vaccines including those for chicken cholera, anthrax, etc. Sorry for the inconvenience. Then another is Emin von Behring and Shibasaburo Kitasato. Both of them induced the formation of diphtheria tetanus antitoxins in rabbit, which were effectively used to treat humans, thus demonstrating humoral immunity. So, they are the two persons who demonstrated the humoral immunity, and this was the basics of the DPT vaccine. Then another scientist, Eli Mechnikov, who demonstrated the existence of phagocytic cells in the blood, thus demonstrating cell mediated immunity. Next is the another important person who is called Edward Jenner. Edward Jenner, who was an English scientist who studied okay, who studied in his natural surroundings in Berkeley, Gloucestershire, and here Jenner was widely credited as a pioneer of smallpox vaccine. So he was the founder of smallpox vaccine, and the findings were very interesting. And he was also called father of immunology. And though at that time he developed a controversial experiment for finding out the smallpox vaccine, that he speculated that becoming infected with cowpox could offer protection against the most virulent smallpox. To test his hypothesis, he created an inoculation with scrapings of cowpox lesions from the fingers of Sarah Nems. It was a young milkmaid, and injected those scrapings of cowpox lesions to an 8-year-old boy named James Phillips. As expected, James developed the mild fever and cowpox lesions, typical of the disease. After a few weeks of recovery, Jenner injected James with the live smallpox virus and found that the boy was indeed protected from the disease. So, these findings he published in 1798 and presented them to the Royal Society of London. Next is about Robert Koch. Not Robert Koch, Joseph Lister. Joseph Lister, who was a British surgeon, was the pioneer of antiseptic surgery, who promoted the idea of sterile surgery while working at the Glasgow Royal Infirmary. Lister successfully introduced carbolic acid because at that times there was an increased cause of contamination during the surgery. So, in order to prevent those contaminations occurring in that infection, carbolic acid was used to sterilize the surgical instruments and to clean the wounds, which led to reduced post-operative infections, and thus it made the surgery safer for the patients. So in 1857, his published findings was transformed the practice. So they are transformed to practice surgery. Next is about germ theory of disease. Once spontaneous generation of microbes was disproved, microbiology exploded. It changed from the observational science to an experimental science. The way was open to study the cause of infectious disease. Building on the Pasteur's work, a German physician, Robert Koch, proved that Microorganisms such as germs, as they were and are still sometimes called the microorganisms, or they are also called germs. They cause diseases. He showed further that specific microorganisms cause specific disease. Koch also introduced higher scientific standards of rigor to microbiology as exemplified by those called Koch postulates. So, Koch, we can discuss now about Koch postulates. Sorry, there is some trouble with the slides. Okay, about Koch's postulates. The causative agent. It is important question, okay? So you should remember the causative agent of the disease must be present in all affected organisms but absent in healthy individuals. Causative agent. And those agents must be capable of being isolated and cultured in pure form. We can cultivate it in pure form. When the cultured agent is introduced to a healthy person, the same disease must occur. The same causative agent can be isolated again from that affected host. So these are the mm, four important and famous Koch's postulates. So this is the time to wind up this session. So we can see at the next session. Okay. Thank you.